Okay, so I've been having a lot of questions about the neuromuscular junction, so I thought I would draw it all out for you guys. This is also the steps of excitation, so the steps that happen before contraction in the muscle cells. Okay, so I'm going to draw this out for you step by step, but first I'm going to give you some players that you need to label. Okay, so here's some of the things I asked you to identify: voltage regulated and chemical regulated channels, active and passive transport, and then there's parts of the cells of the neuron and of the muscle cells, the T-tubule, sarcolemma, sarcoplasmic reticulum, terminal cisterns, triad, and also we could add in here your axon terminal, and your vesicles. Okay, and I'm not sure if I'm going to have space, but these are the ones that you should know. Okay, so we're going to start then. Yeah, we are going to start then with our axon terminal. Okay, so that's the end of an axon, the end of a neuron. This is our axon terminal here. On the other side, we then have our muscle cell. And so all of this together is called the neuromuscular junction. Okay? Alright, so that's the big picture. That's everything. And if you break that apart, you have a neuron and you have a muscle cell, neuromuscular junction. Okay, so the first step in the neuromuscular junction is you receive an action potential from the axon. Okay, the action potential causes a change in voltage, a change in potential. All these things are synonyms for a voltage. On the axon terminal, there are gated channels that, when stimulated by the action potential, cause calcium to flow into the cell. Okay, so this is a voltage regulated channel. And it is also passive transport. If you remember from our diagram, calcium was in greater concentration outside the cell. All this is doing is opening the channels, and then the calcium goes from greater concentration to lower concentration inside the cell. Passive transport, or also facilitated diffusion. Okay, once calcium is in the cell, we have vesicles. I'm going to draw two here, filled with a neurotransmitter called acetylcholine. ACH. I'm just going to put an A. Okay, that is acetylcholine. On the other side of the synaptic cleft, so this space here, is our synaptic cleft. Okay, there's another player we need to identify. Synaptic cleft. Okay, again, the space in between the axon terminal and the muscle cell. It's called the synaptic cleft. Okay, so calcium rushes into the cell. That causes, I'm going to draw a few more of these receptors. Okay, when calcium rushes into the cell, it causes exocytosis of these vesicles, and that then release acetylcholine across the synaptic cleft. So exocytosis is basically the blending of the plasma membranes, and then whatever is inside is released. Okay, this requires ATP, so it is active transport. Exocytosis is active transport. Okay? Once the acetylcholine has been released through exocytosis, it diffuses across the synaptic cleft. Diffusion is passive transport. So release by exocytosis is active transport, but diffusion across the synaptic cleft is passive transport. Okay, the acetylcholine binds to the receptors and that allows sodium to flow into the cell. Okay, that is a change in membrane potential or a change in voltage or a action potential. So these, because you have acetylcholine, which is a neurotransmitter, are binding to a receptor, these are chemically regulated channels. Okay? They are stimulated by a chemical, our neurotransmitter acetylcholine. Okay. So, once acetylcholine causes this signal to be passed across the sarcolemma. Okay, the sarcolemma is the plasma membrane of the muscle cell. Okay, there are extensions of the sarcolemma that go into the muscle cell called a T-tubule. Okay, it kind of makes a T shape. So this is our T-tubule here. Those butt up against the sarcoplasmic reticulum. And the sarcoplasmic reticulum is on the outside of our sarcomere, which is the functional unit of the muscle cell. Okay, the T-tubule then receives that same signal, okay, and passes it along to our sarcoplasmic reticulum. These are sarcoplasmic reticuli. The ends of the sarcoplasmic reticulum are called terminal cisterns. Okay, so this end here and this end here are terminal cisterns. Okay, all together, so all three of them are called a triad. So this end of the terminal cistern, this end of the terminal cistern, and this T-tubule make a triad. Okay, within our sarcoplasmic reticulum, we have our friend calcium. Lots of it. When the signal is transported along the sarcolemma into the T-tubule, you have voltage-regulated channels that cause the release of calcium. Okay, so we had voltage-regulated channels up here on the axon terminal. We also have voltage-regulated channels down here at the triad on the sarcoplasmic reticulum. Voltage-regulated channels cause calcium to be released from the sarcoplasmic reticulum. Okay, this again is passive transport. It's going from higher concentration to lower concentration. Now, inside the sarcolemma, you have actin filaments. Okay. Actin filaments that have complexes of troponin and tropomyosin. Okay, the troponin and tropomyosin wrap around, this is not a very good drawing, but they wrap around the actin. When they receive that signal from calcium, they shift and they expose the myosin binding sites, which then allows contraction to occur. So that allows ratcheting between action and myosin. Okay? When the muscle is done, that shifts back because calcium is then pumped back into the cell. Okay, so when there's no more calcium, that shifts back and it blocks the myosin binding sites. That pumping back in is active transport. Okay, it requires ATP and it's going against its concentration gradient from lower concentration to higher concentration. Okay, so that is almost the whole story. One other thing, to get this signal to stop, acetylcholine will stay bound to its receptor, and that signal will continue to go as long as it's bound there. So, in order to get it to come out, there's an enzyme called acetylcholine esterase. Acetylcholine esterase. And what it does is it breaks apart acetylcholine so it comes in here and it breaks acetylcholine into pieces where it's no longer functional and no longer binds to the receptor. So that stops this signal from being transmitted, which keeps calcium from being released. And then also the calcium being pumped back into the sarcoplasmic reticulum causes the troponin and tropomyosin complex to shift back over, so it blocks the binding sites on actin, so that actin and myosin no longer bind together. These are all the steps of excitation. And then also relaxation. So in order to get it to stop, you have to pump the calcium back in. You have to get acetylcholine to stop binding. And that's it. So these are all the steps. You can identify active transport and passive transport within there. You can identify voltage regulated channels and chemical regulated channels within there. You can identify all the different pieces. And altogether, this is called the neuromuscular junction. Okay, so one thing we forgot to label are our vesicles. Those are right there, which hold the acetylcholine.